Terrific.
All right. Does it look good? Okay. So hello, everyone. My name is Monolina, and I'm a fourth-year psychology major. I serve as a research assistant in Dr. Korzis's behavioral neuroscience lab, and today I will be presenting my research on safety learning and its underlying neural mechanisms, focusing in on an efficient method for identifying artifacts using calcium imaging through a deep learning approach. It's kind of a mouthful. So to begin, I'm going to highlight some key terms that are critical to the study. So safety learning, which is at the core of this project, is the understanding and encoding of behaviors in response to safety and danger to memory. Successful safety learning is indicated by accurate classifications of danger and safety, and unsuccessful encoding of safety learning can result in anxiety disorders and irrational fears. In our lab, fear conditioning is utilized to control and measure safety learning performance. And this learning procedure is a form of classical conditioning in which a subject, in our case, an animal model, is exposed to an aversive stimuli, so something like an electrical shock, and that aversive stimuli is paired with either a neutral context, like a location, or another stimulus like a tone. And after prolonged exposure, fear responses are expressed in response to the neutral context or the additional stimulus alone, without the shock. So this image here highlights the mechanisms of fear conditioning pretty well. In the first image, which is the one on the top, an animal model is shocked while a tone is being played in the background. And in the second image, which is after prolonged exposure, the animal model can be seen freezing after just hearing the tone. Our lab utilizes contextual stimuli, so the use of a location in our fear conditioning, but it yields the same results in which the animal model exhibits freezing without the shock. So fear conditioning can result in fear discrimination and fear generalization. Fear discrimination is the successful differentiation between safety and danger. And this results from the conditioned fear responses. In contrast, unsuccessful differentiation is referred to as fear generalization. In this case, animal models spread conditioned fear responses to similar or related stimuli. So a mouse would freeze in a location that it was not trained in. Fear generalization is a contributing factor in anxiety disorders like PTSD. The data that we retrieve to these conditioning trials is through a process known as calcium imaging in the brains of these models. Calcium imaging measures calcium concentrations in the brain, which serves as indicators of neural activity. We specifically use in vivo calcium imaging, which allows the study of populations of neurons within specific brain regions of freely behaving animal models. So with all of that in mind, I want to discuss the overall goal of safety learning research in our lab, which is to investigate the circuit level mechanisms by which the medial prefrontal cortex supports freer discrimination learning to differentiate between safety and danger, and the mechanisms by which maladaptations in prefrontal circuits lead to neuropsychiatric vulnerabilities. Now, again, that's quite a mouthful, but simply we are trying to identify the location in the brain that supports fear discrimination and fear generalization underlying safety learning mechanisms. This diagram here highlights the experimental design that we follow in this investigation. So in the top left corner, we can see an image of a mouse. And this mouse has a mini scope that is surgically implanted to conduct calcium imaging. And then in the image right below it is a snapshot from a video of the behavioral training I mentioned previously. And then following along to the top right corner, we can see the raw footage that we obtained from this calcium imaging. And then oh, I'm so sorry. Wait, I'm so sorry. Give me 1 second, I'm having some technical difficulties. Sorry about that. And then right below that is the process data that we run through a software called CNMFE. I'm going to talk about CNMFE a little bit later, but it's essentially what prepares our data for analysis. So as I mentioned, CNMFE prepares our data for analysis, but it often includes artifacts in its final output, and artifacts cannot be used for data analysis. And currently, we have a manual method to conduct artifact removal, but it's extremely labor intensive and subject to bias. So this project proposes a computation method that is driven by machine learning mechanisms. So in the study, I asked, can a deep learning classification algorithm in MATLAB be successful and efficient in labeling good and bad neurons in MPFC circuit level mechanisms underlying safety learning? And based on the research I've done on deep learning, I think it can. So the methodology pertaining to safety learning research can be broken down into two components: data acquisition and data analysis. Data acquisition includes the behavior trials that I mentioned, in which animal models are subjected to stages of brain surgery during behavioral testing. This allows for the study of specific brain areas and the neuronal circuits being used during the safety learning. Data analysis utilizes computational techniques. The raw footage obtained from the miniscope is modified, as I said, through a software called CNMFE. And CNMFE is a code that determines the location and quantity of neurons in a video. And after this identification, it extracts calcium signals and creates individual neuron files that are analyzed further. It's extremely helpful in demixing and denoising signals of interest from neighboring cells. But despite being so powerful, again, it includes artifacts. So with some help, I figured out that a deep learning approach may aid in reducing these issues. Deep learning utilizes networks to retrieve useful representations of features directly from data, such that it can be trained to accurately identify and classify images. It's a branch of machine learning that is especially suited for image recognition and utilizes neural networks to train and learn from provided material. There are different categories of neural networks, two of which include convolutional neural networks, or CNNs, and long short-term memory neural networks, LTSMs, but these are pretty difficult to explain because they're pretty complex in structure. So the overarching idea is that deep learning models train with present data to accurately classify future data. This diagram right here is a simplified version of a sequence of code that a model undergoes in a deep learning setup. So before diving into our, wait, I think I skipped some. I'm so sorry for all these technical difficulties. So this developed deep learning network takes files from the raw data of the mice, like I mentioned, and converts them into 21 frames of video to highlight points of maximum fluorescence. It isolates them and then organizes them into their designated categories, which in our case, we wanted to designate good neurons and bad neurons. The general structure of the code is as follows: load training data, define layers, define training options, train, load testing data, and then ultimately determine accuracy. Step 4, which is the training, is critical to the success of the network as it affects the overall accuracy of the model. The network trains with training data, testing data, and validation data. And the breakdown can be seen here on the slide. And essentially, the network trains with 80% of the data values we provide, and then tests itself with 10% of the data values, and then validates itself through further testing with 10% of holdout data. And holdout data is basically data that it didn't train or test with earlier in the process. And this is to steer the model in the right direction. All right, so one more thing right before I dive into results, I just wanted to show what good and bad neurons look like. And so the videos that we used to train our models were predetermined and pre-organized just to ensure that the network was being trained accurately. And the main indicator of what should and should not be classified as an artifact is the presence of fluorescence. And simply, fluorescence is indicated by a bright flash in the neuron. And this can clearly be seen in the good neuron on the right. There are two distinct spikes right there and right there. I hope you guys can see. And then in contrast, we have the bad neuron on the left where there's no fluorescence. And this is what we consider an artifact. For this project, the deep learning model was trained on 4 trials, and trial 1 used 50 neurons, 25 of which were good, 25 of which were bad. And then a second trial with 150, half being good, half being bad. And then 300 neurons with half being good, half being bad. And the last trial was 377 neurons with 187 being bad and 190 being good. And after the training, the models were tested using data that it had not been previously exposed to. So we tested it with a total of 56 neurons, 28 being bad and 28 being good. And finally, we get to the results. So after running training for the 4 trials, each yielded varying validation accuracies, which is the graph on top with the blue lines in each of the images. And validation accuracy provides an unbiased model fit on future performance. So it's essentially like a predictor. So that's what we can see in these figures. Going more in depth, this table highlights the detailed results of each trial. So trial 1 resulted in a model with 100% accuracy. Trial 2 resulted with a model 80%, trial 3 with 67%, and trial 4 with about 74%. The overall accuracy retrieved from each trial is an average of the performances of the classification of good and bad neurons individually. And despite varying degrees of accuracy, the models performed about the same during each trial. In trial 2, however, we can see that good neurons were labeled substantially more accurately than bad neurons. And in trial 4, bad neurons outperformed the labeling of good neurons. And this is pretty interesting given that there were no biases in the setup of our training files. Like I said, we did half good, half bad for each trial. And furthermore, we can see that there were discrepancies between the model accuracy and the test performance, in that the accuracy of the models increased as the number of training neurons decreased. And this is unexpected because deep learning networks theoretically should get smarter with exposure to more data. And it's not like really clear why the model is performing this way, but larger quantities of neurons may exhibit more variation, which may make it more difficult for the network to find a pattern. Furthermore, a lack of improvement in the loss function suggests that the model may be overfitting or underfitting the data. So based on these results that I just discussed, it's evident that the model is learning the training data, but it's unable to generalize it to new data. So we were able to successfully create and train a deep learning network with the present data, but future research should make alterations to the network to allow for successful generalization. So how would we do this? Oh my gosh, I'm so sorry. This is ridiculous, I'm so sorry. So one possible method would be to reduce or increase the number of frames of the videos, and this may allow for more accurate identification of fluorescence. Furthermore, it may be productive to reduce the number of pixels in the neural images to improve both efficiency and accuracy. Alongside the modification, it may be helpful to adjust the parameter settings within the network just to make sure it's not overfitting or underfitting the data. Overall, though, a deep learning network is a complex algorithm that can fundamentally shift how we conduct artifact removal in our research. An accurately developed model can organize and classify neural cells without labor-intensive restraints or biased influences, which can lead to like substantial data analysis. So with that, I'd like to thank the Cortex Laboratory for all their assistance, and I'd especially like to thank Justin Pastor for writing this deep learning code that I presented and for assisting me in organizing this project. And with that, I'll conclude my presentation. If anyone has any questions, I'd love to answer them.
So we've got 2 minutes, not quite 2 minutes left.
Okay.